In this lecture, we're going to talk about the chromosomal basis of inheritance, and this follows our lecture on Mendel. So just a little bit of history. Mendel, recall, was doing his experiments with peas back in the 1850s, and this is when he came up with his two laws that are still form the basis of genetics today, laws of segregation and independent assortment. We'll review those shortly. And then notice that about 30 years later, mitosis and meiosis were described. So keep in mind that Mendel came up with his findings long before they knew about DNA, chromosomes, or meiosis. Shortly after meiosis and mitosis were described, the chromosomal theory of inheritance was put forth, which basically says that Mendel's hereditary units are located on chromosomes. And this logically followed the characterization of mitosis and meiosis, since that's what's being moved during those processes, the chromosomes. And then notice it took approximately 50 years after the chromosomal theory of inheritance to actually ascertain that it is DNA in the chromosome and not the other components, the other main one being proteins, that is serving as the genetic material. So we'll be covering DNA in Unit 4 in more detail. So just to remind you about Mendel's two laws, his law of segregation is based on the idea or the fact that for different genes that organisms possess, there are two alleles, one inherited from each parent. And so we say that this organism is diploid. It has a pair of alleles for a specific gene. And then during meiosis, the allele pairs are segregated into different gametes. So these are the gametes here. Notice they have either only a purple or only a white allele for flower color, not both, because gametes are haploid. This is the whole point of meiosis, is to half the number of chromosomes, and not just any half, but one of each homologous pair. Because as we've discussed many times, these alleles for a gene are located on homologous chromosomes. We'll review that as well. So this is Mendel's law of segregation. The two alleles for a gene are segregated during meiosis into separate gametes. And then when you make your Punnett square, you put the gametes for each sex across the top and down the side of your Punnett square. And it doesn't matter, egg or sperm, either one can go on the top or down the side. Okay, and then Mendel's second law, the law of independent assortment, has to do with alleles for different genes. So if we look at the gene for seed color and also for shape, like round versus wrinkled, yellow versus green, notice that if you have two parents, one yellow round, one green wrinkled, and their offspring, their F1 generation, is all yellow and round, first of all, that's how you decide if it is a dominant recessive trait. Then this is how you decide which is the dominant allele. If the F1 are yellow, even though they have a green allele, then yellow is dominant to green, or we say green is the recessive allele. And then the same with round versus wrinkled. Round is dominant over wrinkled. How do we know? Because the F1 is round, even though they have a round and a wrinkled allele. So if you cross two F1s and look at the F2, notice the different offspring that are possible. It's not just offspring that look like either well, that are round and yellow. You can also get some round green, some wrinkled yellow. And this is what Mendel based his second law on, law of independent assortment. Alleles on non-homologous that are not homologous or not on the for the same gene will segregate independently of others during meiosis. Here we see, we've looked at this many times, this shows a homologous pair of chromosomes, and here are the alleles located on the homologous chromosomes and in the same locus or location. And once again, during meiosis, a homologous pair, shown here as the red and blue, are going to be segregated or separate during meiosis, and the gametes that are formed are just going to have one of the homologous pairs. So they're just going to have one of each allele pair for a gene. Here we see a gene that's been fluorescently stained, so you can see that this is a chromosome that's in the duplicated stage, so you see 2 copies of it. Okay, and mainly we want to focus on this part in green here. This is the chromosomal theory, really very straightforward, stating that Mendel's hereditary units are located on chromosomes. Okay, and here we see that those non-homologous chromosomes can line up on the metaphase plate in various ways. How exactly the non-homologous pairs line up is random, so we call this independent assortment at metaphase 1. We talked about this back when we discussed the various ways that genetic variability is increased. So I would like you to recognize this name, Thomas Hunt Morgan. He is credited with providing proof or support for the chromosomal theory of inheritance, and he also introduced one of the, to this day, popular organisms to use for studying genetics, the fruit fly, Drosophila. The fruit fly is good, is a good model because here are some of the great traits. Fruit flies, they reproduce at a high rate. They can breed a new generation every couple weeks. They have only 4 pairs of chromosomes, so 8 chromosomes, 4 homologous pairs. And they have traits that you can follow, like eye color. And that's actually what Thomas Hunt Morgan used, was eye color in Drosophila to give support to the chromosomal theory. And so his luck was that he found in all of his fruit fly breedings some mutants, because often mutants hold the key they provide information. So he found a white-eyed mutant male. So here we see a Drosophila eye. This is not normally they have a red color. And I'm going to use the symbol that Thomas Hunt Morgan introduced that is still used when doing fruit fly genetics. So if any of you go on and take genetics, I'm sure you'll work with this. So W with a positive sign, this represents the wild type phenotype, in this case red eyes, and then white eyes would be symbolized with just a W. Okay, so let me just briefly talk about how Thomas Hunt Morgan showed that genes are in fact located on chromosomes. So what he did was that he obtained he obtained some F1s that were heterozygous for red eyes. So he had he took red-eyed females who had a red allele and a white allele, but since red is dominant to white, they had red eyes. And then he took what he thought the male genotype was. It also had red eyes, so he thought it would have the same, genotype. These were the F1s. Okay. So when he did the cross, and let's just set up this Punnett square here. So you've got the two gametes across the top and down the side. Fill in your Punnett square. Try to be neat about it. So, but once again, what's across the top are the genotype of the gametes of this parent. And then down the side is the genotype of the gametes of the other parent. And in this problem, they happen to be the same genotypes. So we're going to have, let me go back to here. All right, so if this gamete and this gamete undergo fertilization, this would be the genotype of the new offspring. So I'm just going to fill in the Punnett square. I apologize, it's a little messy. And then we'll analyze it. So what he was expecting when he crossed these two heterozygous red-eyed male and female fruit flies is he was expecting that 3 out of 4 would have red eyes. See, this is a homozygous red-eyed, and then you expect 2 more to be also red-eyed but heterozygous. So that's 3 out of 4 red eyes, or 75% chance red eyes, and then a 25% chance that the fruit flies would have white eyes. Okay, and that is what he saw. That is what he found. Except, and here was the unusual thing, is that normally if this was the case, you would expect 25% of the offspring to have white eyes, and it would be roughly 50-50 whether they were male or female. But what Thomas Hunt Morgan found was that 100% of the white-eyed fruit flies were male. And so that is what led to the conclusion that the way that was possible was that the gene for eye color in fruit flies is actually on the X chromosome. So the female had 2 X chromosomes, one with the red and one with the white allele, and then that they were mated with the male who had red eyes, so he had the red allele and then the Y chromosome. This is a male. So in this case, when you set up your Punnett square, we'll put the female across the top and the male down the side. So some of the male gametes will have the X chromosome with the red eye allele on it, and the others will have the Y chromosome. And in this case, and remember, when you're doing sex-linked problems, you include the X and the Y chromosome. So you're going to keep track not just of the alleles, but of whether they're of the X and Y chromosomes. So in this case, when we fill out the Punnett square and analyze it, we see that all the females are red-eyed. X-axis female. So they're all red eyes. Yes, they have different genotypes. One's homozygous red eye, one's heterozygous, but they're all red-eyed is their phenotype. And then for the males, half have red eyes and half have white eyes. But if we're looking at the entire probability here based on the Punnett square, 25% of the offspring are expected to be white-eyed males. And that is what they observed in their lab. And thus, this was the first time that it was definitively shown, or a link between an allele and it being located in this case on the X chromosome. So hence this supported the chromosomal theory that genes are located on chromosomes. Oops, sorry about that. Okay, so sex chromosomes, once again, are in when we're talking about mammals, the sex chromosomes are referred to as X and Y, and I think you already know this, but just to make sure, females are XX and males have one X and one Y. There is a term for that, hemizygous. Now, here's a scanning electron micrograph of the X and Y chromosomes. They are considered homologous, remember, although they are different from your normal generic homologous chromosomes in that they do not contain genes for the same traits. I mean, that's pretty obvious just from looking at their size difference. But together they determine genetic sex. So they're considered a homologous pair. Now, not all organisms use the XX for female, XY for male method for sex determination. So we're going to take a very brief foray into the different types of sex determination. So we already talked about the XY mode in mammals. You also see it in some insects and some plants. But there's also cases where the difference between male and female is diploid versus haploid. So this is the symbol for female right here. So notice that in grasshoppers, and this is true for some other insects, the females are, they have two X chromosomes. So they have two copies, but the male only has one. So that's one way, another way to determine sex. Or there are also cases where it's the female that has the two different alleles, whereas the male is homozygous. So they call that the ZW mode. And then here's where the entire organism is diploid versus haploid. So the female has twice as many chromosomes as the male. Okay, so those are some other examples, just so you don't think it's always XX female, XY male. So once again, if a gene is on the X or Y chromosome, we call it sex-linked. And in a genetics problem, you're probably going to be told something is X-linked, because you do need to know if it's sex-linked, which chromosome it's on. Is it the X or the Y? So there are some disorders that are recessive diseases, meaning you need two copies of the defective gene, or if it's recessive for a male, you just need one copy on the X chromosome. So here's some examples: colorblindness, muscular dystrophy, Duchenne muscular dystrophy, hemophilia, which is a blood clotting disorder. These are X-linked genetic diseases. We'll be doing some examples during lab on Thursday. So what about the fact that females have two X and males only one X chromosome? Does that mean that females have, in overall, more functional alleles than males do? Well, the answer basically is no, because in females, one of the X chromosomes in the female cell becomes permanently inactivated. It's called X inactivation. And it's like the chromosome condenses, and none of the genes on that chromosome are used. So it's inactivated. And this happens fairly early on in development in the embryo. And it is random in that in some cells in the embryo, the X chromosome from the mother might become the inactivated ones. And in other cells, it might be the X chromosome that was inherited from the father. And so you can really see and it's called a bar body. That's what you call it after it's inactivated. So it's highly condensed, inactivated. And this is only going to happen in females, right? You don't want this to happen in males because then they won't have any X chromosome. And there's some very important genes on there that males and females need. Okay, here's a nice example illustrating X inactivation. In this type of cat, tortoiseshell cat, the color for their coat, whether it's black or orange, happens to be on the X chromosome. So that's convenient for kind of visualizing X inactivation because early on in the embryo of this kitty here, in some cells, the X chromosome with the black allele becomes inactivated, but in other cells, it's the one with the orange allele. So some cells express the orange color and some the black. So you can see if you look at this cat's coat color, it's hard to say, you see a mixture of black and orange. So we call this tortoise shell, and it's only going to be in females, right? Because the male only has one X. They're not going to inactivate it. This is a nice little overview of X-linked alleles and how they're passed on to offspring. So if you're talking about a genetic disease that's recessive, where like if capital A is the normal allele and little a is the defective gene for something like hemophilia, then this shows you all the different possibilities based on the parent's genetics. So for example, in example A, if the male carries the defective allele, so if we're talking about hemophilia, for example, this male would have hemophilia. And then if the female, if the mother is completely homozygous normal without any defective alleles, then these are the possible offspring they can have. So notice that none of their children are predicted to have the disease, if that's what we're looking at. However, their girls that they have will be carriers of the defective allele that was on the father's X chromosome. So, the problem could come in the next generation when they have children. Okay, and then in the middle, let's look at here we see normal sperm, but with a carrier female. So in this case, notice that if they have children, they have a 25% chance of having a baby, and in fact it would be a boy, with that genetic disease. So again, if we're talking about hemophilia, they have a carrier mother and a normal male can have a 25% chance of having a baby with hemophilia, and it's going to be male. So this is why X-linked recessive genetic diseases are seen more in males. Because it's recessive for females, even though, look at this daughter here, she also has the defective allele. So why doesn't she have the disease? Because she has another X chromosome with the normal allele, and that's enough to prevent. She will not have the disease. We call her a carrier. And then in this third scenario, we have a male with the disease with a female carrier. And in this case, they have a 50% chance of having a baby, either boy or girl, with the genetic defect. So it's for things like this that people sometimes have genetic counseling done. If there's a history of hemophilia or muscular dystrophy or something in one's family, then you might want to have some tests done. Okay, so we talked about independent assortment earlier, how non-homologous, how genes for different traits can assort independently during meiosis. But what happens if those genes are on the same chromosome? Or can they still end up in different gametes if they're on the same chromosome? basically the answer is yes, they can still end up in different chromosomes because of crossing over during prophase 1. So it is possible, but it is less likely to happen than if they were on, let's say, separate chromosomes or far apart on the same chromosome. And so we say that genes on the same chromosome are linked. And the closer together they are, the more linked they are. And the more linked they are, the more likely they are to go together into the same gamete. In other words, to be inherited together. So linked genes, here it is, tend to be inherited together because they're located, near each other on the same chromosome. So we call those linked genes. Okay, so Thomas Hunt Morgan, not only did he was he sort of famous for proving the chromosomal theory, but he also did a lot of work that helped map the location of genes on chromosomes. We're not going to be covering this in this course in detail, but I did want to mention it, that before they were able to actually sequence genes and locate pretty definitively many genes on chromosomes, they use these kinds of linkage experiments where Morgan and his colleagues would look at certain inheritable traits like eye color, wing length, body color, antenna length, things like that. And they would see whether or not these different traits tended to inherit together or not. And the more often you say, let's say, a black body and a short antenna were inherited together, the closer it was surmised correctly that the closer together they were located on the chromosome. So by looking at this kind of data, Hunt Morgan was able to make linkage maps, which gave you a somewhat crude, but still, an idea of where these different genes are on the chromosomes. Okay, now the last topic I want to cover today is talking about chromosomal abnormalities. What happens if you inherit an abnormal number of chromosomes, like too many or too few? So let's look at that briefly. So one thing that can go wrong during meiosis is something called non-disjunction, which means that the chromosomes are not separating properly during meiosis like they're supposed to. This can happen, let's look at this picture. This can happen in meiosis 1 or in meiosis 2. It can happen in both stages. Let's look at meiosis 1 for a moment. Remember that in meiosis 1, that's the big event, is that the synapsed homologous chromosomes are going to separate and be pulled to opposite poles of the cell. And if they fail to do that and a homologous pair goes to the same pole, then it's going to end up in the same, they'll end up in the same gamete. And you'll have some gametes, these are the 4 daughter cells here, you'll have some gametes with an extra chromosome. N is the normal haploid number that you would expect, plus 1 is an extra chromosome. And then what about the other pole where there was no homologous pair that went to it? So you're going to have some gametes that are minus 1 chromosome. So if we're talking about humans, human gametes have 23 as the N number. So these would have 24, or some would have 22 chromosomes. Okay, so that's non-disjunction. And here's on this side, you can see it can also happen in meiosis 2 when the sister chromatids are supposed, the centrosome is supposed to break and they're going to travel to opposite sides of the cell and then end up in different cells. Well, that can also fail to happen. And so again, you can get a gamete with an extra or one lacking a chromosome. So non-disjunction leads to abnormal gametes in terms of chromosome number. And the problem arises if any of these gametes end up fertilizing or being fertilized. And then the new zygote will have an abnormal number of chromosomes. So aneuploidy is the term they use. Abnormal number of chromosomes. So for example, here's one of the most well-known, Down syndrome. And in this is a case caused by non-disjunction. And look at chromosome pair number 21. Notice there's 3 of them instead of 2. Should be just 2 homologous pairs. So this little girl has an extra chromosome. And you might think, well, what's the problem with that? Just more genetic information. But it actually interferes with regulation of gene expression. And Down syndrome comes with some pretty serious effects, health effects, and reduction of mental capabilities. So it's an example of non-disjunction. They call it trisomy because there's 3 copies of the chromosome, number 21. So here are some of the malformations. People with Down syndrome tend to have circulatory problems, mental retardation. Life expectancy is not really that high compared to the current life expectancy for a healthy adult. And it's, somewhat regular. I mean, it's more common as the parents get older. So it's not really, Down syndrome is not a genetic disease that you would pass on to your offspring. It's a failure of meiosis to occur properly. And that is why we talked about this before, but just to remind you, checking for the right chromosome number is one of the main objectives of amniocentesis or CBS sampling, both done to obtain some of the baby's DNA, either by injecting a needle into the amnion and aspirating out some skin cells that are sloughed off the baby's skin, and then from there obtaining DNA, or from getting a snip of some tissue that's going to form the placenta that actually comes from the zygote, so it has the same DNA. So that's called CVS, chorionic villus sampling. Anyway, they both have the same objective, to check for chromosomal abnormalities. Okay, and I want to stress that this is a genetic analysis, genetic test. It's not testing for other metabolic disorders, or it's a for checking the baby's chromosomes. Okay, and then one more topic about chromosomal abnormalities. This slide shows some other things that can happen to chromosomes that can lead to problems. So chromosomes can have a piece of them cut out. That's called a deletion. So now this chromosome is going to be missing some important, maybe they might be very important genes that it's missing. Or sometimes this can happen, for example, when the chromosome is duplicated. Remember, in the S phase of the cell cycle, the chromosome gets copied. So this is one time, just an example of when something like this might happen. A duplication is when a piece of the chromosome accidentally gets copied twice. So you have twice the number just for those genes. Okay, that can also lead to problems. That's called a duplication. There's something called an inversion, where a piece of the chromosome, it's as if it was cut out and then flipped and put in backwards. So that's called an inversion. That can also lead to problems with regulating the genes that are on these chromosomes. And then reciprocal translocation oops, sorry, the lines through there, is when pieces of two different chromosomes are detached and then they are reattached to the other chromosome. So they get translocated to each other. So these are all types of mutations or chromosomal abnormalities that are seen. So for example, just to give you one, there's this syndrome called Cri-du-Chat, which is a deletion in chromosome number 5. So you can see that in this picture. Look at this. This is chromosome pair number 5. You see how this one on the right is shorter? Okay, because there was a deletion there. So this person is missing some genes, and it actually causes very serious defects. They're severely mentally handicapped and usually do not have a very long survival. So here's a little boy who has it. You can see there's some abnormal head shapes and shape of his head, and the name comes from Cry of the Cat because they tend to make a sound like that, like a crying cat. Okay, so that's just one example of a chromosome abnormality disease. Okay, and then this is the last thing I want to show you. Some of these, going back to sex-linked for a moment, I just wanted to remind you, because you have a test coming up, that while we didn't cover pedigrees, I told you, but I want to remind you that there may be a bonus question about pedigrees or using pedigrees on your upcoming test. So again, I'm just reminding you that squares are males, circles are females. If it's shaded in, it means they have the condition, whatever you're talking about, like hemophilia. And if there's a dot inside, it means they're carriers. So like this female is a carrier, this male does not have the disease. And then down here it shows their children. They have 2 boys and a girl, and one of their sons has the disease, the other does not, and neither does the girl. So look that over, see if you have any questions. All right, so that's completes the chapter 15 on the chromosomal theory of basis for genetics.